Alright, we are going to talk about the citric acid cycle. So if you haven't watched my video on glycolysis, go and watch it. That's the first step in this series. After glycolysis, we're going to have the citric acid cycle, and this is going to be where we really expel all the carbons that we split up in glycolysis, and we make them into carbon dioxide. And then later on, not in this video, we'll go over oxidative phosphorylation, where we actually make most of our ATP. But in order to start the citric acid cycle off, we need something called acetyl-CoA. So first we have to transport pyruvate into the matrix of the mitochondrion, because we did glycolysis in the cytoplasm, right? But the mitochondrion is the powerhouse of the cell, and that's where the rest of the process happens. So we're going to take pyruvate via a shuttle into the mitochondria, and inside the mitochondrion we're going to have a little reaction before the citric acid cycle called pyruvate oxidation. And this is going to be done by an enzyme called the pyruvate dehydrogenase complex. And this one is kind of complicated. It's got lots of components. You're going to make pyruvate into something called acyl TPP using a chemical called TPP to attach it to. And this is going to release carbon dioxide. After this, we're going to make acyl lipoate. And acyl lipoate goes through some reactions, and we're finally going to bring in coenzyme A. It's written as CoA-SH because it is attached to a thiol group, and where that thiol group is attached is going to end up where the acetyl group, which is the 2-carbon group from pyruvate, is going to attach. And out of this process, with these many reactions within the pyruvate dehydrogenase complex, we are going to use an NAD+ and reduce it to NADH, because our pyruvate is getting oxidized, right? Every time we oxidize something, we have to reduce something else. And, of course, we're going to get out carbon dioxide. And that is not all the carbon dioxide we're going to get out of this video, but it is the first. So now that we have our acetyl-CoA, we can begin the citric acid cycle. So the citric acid cycle is going to start with something called oxaloacetate. And from oxaloacetate, we can add on acetyl-CoA via an enzyme called citrate synthase, and we're going to get citrate. And you can see all the chemical compounds in this diagram. I'll try to scroll to make this convenient. We'll see how this works. But anyways, citrate is going to go through an enzyme called cis-aconitase, and this is going to make isocitrate. And here, once we have isocitrate, we are going to do our first reduction of the citric acid cycle. Remembering glycolysis and pyruvate oxidation, we were getting these electrons and these protons for oxidative phosphorylation later on in the form of NADH. So we're going to do this again. We have NAD+, and we are going to make NADH in the process of making alpha-ketoglutarate out of isocitrate. This process is also going to release carbon dioxide, since isocitrate is 1 carbon more than alpha-ketoglutarate is. And this is all going to be facilitated by an enzyme called isocitrate dehydrogenase. So we're going to take this alpha-ketoglutarate, and we're going to do this whole NAD+ reduction thing and CO2 release again. And this is done by alpha-ketoglutarate dehydrogenase. So these two reactions are quite similar, and we are going to get something called succinyl-CoA. So we are bringing coenzyme A into the game again for just one step, and then we're going to take it out via an enzyme called succinyl-CoA synthetase, and we are going to make succinate. Now, sometimes enzyme names are going to be backwards, and that just means the process is reversible. So, like, remembering glycolysis when we had pyruvate kinase, and pyruvate kinase removes a phosphate from pyruvate? That's actually because it can add a phosphate or not removes a phosphate from pyruvate, removes a phosphate from the phosphorylated pyruvate. Anyways, the reason why that is we can go in reverse and start with pyruvate, the product, and phosphorylate it via pyruvate kinase to go backwards in that reaction scheme. So it's the same here. Succinyl-CoA synthetase can go back the other way in the cycle and makes acetyl-CoA. But here it is going the other way and going from succinyl-CoA into succinate. And we're going to make something called GDP, or guanosine triphosphate. And this essentially is the same as ATP. If you learn about G-protein coupled receptors, if you learn about monomeric GTPases such as RAS, you might know that GTP can actually activate things in cell signaling, kind of like the way that ATP can. So this one, it's also energy. That's why we care. Succinate is going to get made into fumarate via succinate dehydrogenase, and this is going to make a different electron acceptor, a different little shuttle bus to carry our electrons to the electron transport chain, and this one is called FADH2. So we start with FAD, and we get FADH2. And from fumarate, we have fumarase, the enzyme that's pretty easy to remember, and fumarase is going to bring us to malate. And then malate dehydrogenase is going to make us another NADH and put us all the way back to oxaloacetate, and oxaloacetate is going to take another acetyl-CoA and make citrate all over again. So I know this cycle looks kind of complicated. Chances are you will not have to memorize this whole cycle. Whether you're in high school biology, whether you're in an introductory college bio course or AP Bio, even on the MCAT, I have never seen a question on the MCAT that makes you like spit out these enzyme names without giving you hints from passage. So please don't get too overwhelmed by this big crazy cycle. But we're going to talk a little bit about the ways in which this is regulated. So a lot of the regulation stuff in the citric acid cycle is really common sense. It's going to be feedback inhibition, it's going to be negative feedback, or it's going to be positive feedback the other way around. So either you're going to have products that are inhibiting the enzyme, or we are going to have reactants that are stimulating the enzyme. So for example, pyruvate dehydrogenase complex, the thing way back in the beginning that made pyruvate into acetyl-CoA, this is going to be inhibited by high levels of ATP, by high levels of NADH, and those two things make sense, right? Because the whole point of doing this whole cell respiration and the citric acid cycle in particular is to get ATP, well, the equivalent, which is GTP, and NADH. So that makes sense, right? Products inhibit the continuation of the reaction. Acetyl-CoA, also that one, that's pretty intuitive. Acetyl-CoA is going to inhibit the making of acetyl-CoA, because let's say that instead of doing the pyruvate to acetyl-CoA, we did beta oxidation, which, if you don't know, is the way that we break down fatty acids, and we break down fatty acids into acetyl-CoA. Let's say that we had like a really fatty meal and we broke down all these fatty acids into acetyl-CoA, and we had a whole bunch of acetyl-CoA that can go into our citric acid cycle. Is it useful for us to continue making pyruvate into acetyl-CoA, or can we take that pyruvate and undergo gluconeogenesis and make us some sugars that we can store? We should do the second one, and that's why acetyl-CoA is going to inhibit this pyruvate dehydrogenase complex. The next kind of checkpoint on this, and I've labeled all the places where it's regulated with these little purple triangles, is going to be citrate synthase, and citrate synthase is also inhibited by ATP and NADH, but it's also inhibited by citrate. Again, that makes sense because we're making citrate, and if we have enough of it, we don't need to make any more. And it is inhibited by succinyl-CoA, and succinyl-CoA is like halfway across the cycle, but it follows that if we have a lot of citrate, we also will make a lot of succinyl-CoA. So that pretty much makes sense. And again, likely if you come across a multiple choice about this, none of the other choices will really fit. It will be like, does oxaloacetate inhibit citrate synthase? And then it's like, no, because oxaloacetate is what makes citrate. So that would stimulate it if it was a part of the regulatory system, which I don't think it is. And then there's a couple more. So we've got isocitrate dehydrogenase. This is inhibited by ATP and NADH, and it is stimulated by ADP and NAD+. So that one makes a lot of sense, right? And then we have alpha-ketoglutarate dehydrogenase complex, and that one, again, like all of these regulation mechanisms we've seen, high ATP, high NADH, this one doesn't work as well. This one is also going to be inhibited by succinyl-CoA. So if we look back at this cycle and we have alpha-ketoglutarate, this is going to make succinyl-CoA. So this makes sense, right? If you need to take time to look over these regulatory mechanisms, go back in the video, screenshot them, put them in your notes. But in the end of this whole thing, we are going to get 6 NADHs, 2 FADH2, and 2 GTP, and this is for 2 turns of the cycle, because if we start with 1 glucose, we get 2 pyruvate, right? 2 pyruvate, 2 acetyl-CoA, 2 acetyl-CoA, 2 citrate, and so on. So we are going to get twice everything. Okay, so, and then we're also going to get some CO2s. But those really aren't useful to us. So with all these NADHs and all these FADH2s, they are going to go to the electron chain and they're eventually going to end up splitting back into NAD plus and some electrons and a proton. And same here, we're going to get the FAD and then some electrons. I think it's 2, it might be more, but the point is electrons and a proton, or 2 protons, I think, yeah. So this might be 4. Anyway, so we also get GTP, which goes into cell signaling pathways like the G protein coupled receptor type things, where you have the monomeric GTPase and it will break it down as it's activating itself, or deactivating itself, sorry. But those are the products of the citric acid cycle, and next time I'm going to cover the electron transport chain, and we're going to see how this whole thing that I just put in blue works, and why we want those electrons and why we want those protons to come out of these compounds. I hope this helped.